How does coffee work? Why do we feel less sleepy? Most people think coffee gives us energy, but this is not really the reality. I wanted to use this opportunity to better understand how coffee works or caffeine works inside our body, and at the same time explain and show to the scientists the power of the new version of Vicena 3.5. So why coffee works and how coffee works. These are the two molecules of relevant to this journey. On the left is the caffeine, the molecule inside coffee, and on the right is adenosine. What is adenosine? Is the molecule that is generated or created inside our body. Adenosine is the side effect or the result of consumption of energy in our cells. In our cells, we have these ATPs, these molecules, that they are like a currency of energy inside our cells. Think of it as a molecule, battery molecule for our cells. Every time that we consume energy, we lose one of these phosphate groups. So ATP converts to ADP and AMP and then adenosine. So you see that these phosphate groups removed gives energy, and then the concentration of these adenosines shows how much energy was consumed in our body, how much work it has been done in our body. So when we do activities, when we think, when we study, when we go to do sports, the more we do activities and the more energy is used, more saturation of adenosines in our body increases, or concentration of it increases, and we feel more tired. So, how does this work? Let me first understand the mechanism or the simple mechanism, molecular mechanism of feeling tired. So, these adenosines, they fit into these receptors. These receptors are the membrane receptors, A2A receptors inside the cell walls, membranes. And these adenosines get essentially trapped here in this pocket. And as you see, this is towards the outside of cell, and this part in the bottom is the inside of the cell. These molecules or proteins, which is essentially the sensor inside the cell, they have two states, the active and inactive. When adenosine is outside, they are inactive. When it goes inside, it deforms this molecule and it goes to an active shape. So they have two shapes. So when it goes inside, think of it like essentially the key inside the lock, and it opens the lock. So how does it work? These two have two shapes. In these two versions, they have a different shape. They are deformed. So here on the gray is that when is inactive, and on the colorful like red and blue is when it's active. It has 16 angstrom deformity. 1 angstrom is 0.1 nanometer. So 1/10 of a nanometer. And this is very cool because the molecule gets inside here, let me show you here. The molecule comes here and is a local interaction, but the deformity is global interaction. So it deforms globally, which means essentially it amplifies the effect of that molecule. And this is how it works. And all of these are not visualizations, just the drawings, but the actual computations that is done, if you believe it or not, by me with the help of Vicena. So we have this adenosine and A2A receptors, and when they go in, they create 12 contact points. This sucks and clamps, the receptor clamps around adenosine. As you see, it deforms it with the highest margin, like for example, the swings or reshaping with 16.4 angstrom. Now, let's look at caffeine, the main molecule that we are talking about. As you see, caffeine is kind of similar, but not kind of similar. So they are different. So they have the same part, more or less the key part, that it fits in the same pocket, but it has a different shape, which means that what happens? Let's think the moment that I drink coffee. Let's do a calculation. Let's do a pharmacokinetics calculation and see that does the concentration of caffeine increase in my body when I drink coffee to a level that is relevant for my receptors. So here, as you see, after around an hour, the maximum concentration of caffeine in my body exists in my blood. And this concentration is around micromolar level, which is relevant in competing with adenosines. So if it's very, very low, adenosines, like the receptors, would not, like really not compete. If you look at this, also because caffeine looks different but similar, still the receptor prefers the adenosine over caffeine. And this is relevant. Still, adenosine head-to-head wins and locks in first if you have 1-to-1. But because you have now two molecules competing on the same receptors, then you will have essentially some of these receptors capture the caffeine. And this is how it goes to the same pocket and to the same place. And this is a calculation that shows and just figures out where the docking or goes to the same place. And the cool thing is, this means that now you have some receptors, A2A's receptors, that are occupied by adenosines and some by caffeine. And as you see here, if you increase the concentration of caffeine, the percentage of A2A receptors that are occupied by adenosine decreases. It's simple. You had one choice, now you have two choices. And now essentially it's being blocked. Caffeine, it doesn't deform as much the A2A receptor, which means that it's occupied, but it is not deformed. So the signal is not transferred inside the cell. So inside the cell does not see it. So now you're blocking essentially the signaling pathway of tiredness. So the cells cannot see or feel that there were a lot of energy being consumed and a lot of adenosines being released inside the cells. So when you drink coffee, you are actually blocking the receptors of adenosines, which means you're not generating energy, you are just masking feeling tiredness because your body does not have a way or less receptors receive the signal. And this is how it works. And all of that is thanks to the work that Vicena did in one place for me. I could do literature review. I could understand about receptors. I could do the simulations, and compared to ChatGPT or all the other ones that explains you in words, here I could do primary research. Not only I could read articles, but actually I could generate these results. I will also put this data somewhere that you can download. And I want to give a shout out to essentially the guys at Rowan Scientific, because behind the scene, now Vicena is doing simulations there. And if you have a simulation software or a way that you want us to connect Vicena into, let us know. go and do something cool, go and understand something nice, and I hope that I could explain this in an understandable way. And if you are yourself a scientist and you saw I made some mistakes, please let me know in the comments. Thank you very much.